to be talking to you guys about what transcription is. So transcription is in our body is like the first step of protein synthesis. So with transcription, the reason why we have it is because DNA can never leave the nucleus. So for that reason, we need to transcript some codes to make proteins and other stuff. So those codes need to leave the nucleus so we're able to make the proteins. So let me start off explaining. This is the 3' end right here. So everything is read from the 3 to the 5' end. After reading everything to the 3' end, we have an indicator or signal or promoter those are all different names that can be used for it, that comes down and latches on right here. The reason why it latches on right here is because that's where the data box is. We don't use this is the only area that we're going to use for this code. We don't use the whole strand, because over here is a different code for some other protein that we don't need. So what a data box is, to explain that, is a series of T's and A's on the strand. So after that, a helicase enzyme comes in and breaks up the hydrogen and nitrogen bonds. So as you see, we have this is now called an open complex. So we have this open complex with this little slice right here, or opening right here, which is where the RNA will be made, or right now, which is called the pre-mRNA. So this is a pre-M, pre-mRNA right there. So everything right now, okay, so if we look right now, we have that RNA polymerase right here, this blue kind of looking or greenish looking sac thing. So it transcripts or reads everything from the 3' end to the 5' end. So everything is read from the 3 to the 5. And then we have what is called the 5' end right here, prime end, which is an anti-parallel, which starts transcripting or synthesizing from the 5' end to the 3' end right here. So all right here are genes or codes right here that are being made by or attaching and being made from the nitrogen bonds down here. And over here, it has to stop somewhere, so we have a stop codon right here and where it stops at. So basically, if we were a prokaryotic cell right now, everything would we would basically be done. But since we are more complex, or eukaryotic cells are more complex, we're not done, and that's why it's called the pre-mRNA. So I'm gonna go to another picture to explain what happens next. So, let me go back. So you remember our 5' and 3' end? So before this code can leave the cell, we have to prepare it to leave. So we attach on the 5' end right here. It's called a 5 cap. So these little circle little things will attach on right here. It's called a 5 cap. And then over here, pony A tails will attach onto the 3' end over here. So little tails over here will attach down here. So on the 5' end, just to tell you, a 5' end cap is placed there, and a pony A tail is placed to the 3' end. And now I will switch to the other picture. So this would down here would be let me get a brush, sorry. Down here would be the 5' end cap. So the 5 cap. And down here are the pony A tails. I'm just gonna abbreviate it with tails. Oops. Yeah. which I abbreviated. So what happens is, well, the reason why we have these 5 end caps down here is because when we leave the nucleus, we don't want to, like, all the stuff inside is very important, and we don't want to lose anything. So if this, like, traveling, going outside the nucleus, because this is the only thing that can leave the nucleus, because this is a copy of a DNA, it's not the actual DNA. If we have, like, it going through, like, moving outside, and, like, one of these falls off, that's okay. And when, like, we have so many of the pony A tails, so if, like, it gets banged around and one falls off, that's okay. That's the reason they're, like, bumpers, to protect the important stuff inside, the codes inside. So I'm gonna go back up here to this diagram. So before it can leave the nucleus, we have, like, these introns, which are, like, the codes that we don't need. So what occurs is splicing occurs, so, like, it takes out the stuff we don't need before we leave. So that's what happens, like, this splicing occurs to eliminate, like, introns, because exons are, like, the code readers, and those are the only ones that can, like, leave and are, like, helpful when they leave. So that's why we have only exons down here, because splicing occurs. And then this like, pre-mRNA strand up here, like, after splicing becomes RNA. So that's why in the other picture before I wrote pre-mRNA, because nothing was spliced off. So basically, this is what transcription is, and thanks for watching.